So good morning, good afternoon, ladies and gentlemen. First of all, I would like to thank you for inviting me to this webinar. And in the next half an hour, I would like to speak about a little bit on G-protein coupled receptors and more specifically on the remote controlling option of the receptor function from the extracellular vestibule. Just because maybe not all are familiar with the GPCRs, then I would like to shortly introduce this pretty much important target class that gave already many successful drugs. Actually, 30% of the FDA-approved drugs are targeting GPCRs. The total number of 481 FDA-approved drugs targeting 107 unique GPCR proteins. However, this is not the whole number of the GPCRs we know, because this is the typically large target family with very significant pharmacological relevance that includes 375 different targets, out of which 225 are known endogenous ligands, and about 150, these are orphan receptors having no knowledge on the endogenous ligands. So there are different motivations why GPCRs are especially important, and I would like to mention further two motivations. One is the many opportunities available for the modulation of the GPCRs, and these are basically new opportunities that includes the use of multiple signaling pathways, and in addition to the orthosteric binding sites, we can address different allosteric binding sites available on GPCRs, and there are also oligomerization and dimerization processes with significant pharmacological effect that we can also modulate somehow. So these many new opportunities have been exemplified by the Chemistry Nobel Prize 2012, awarded to Rob Lefkowitz and Brian Kobilko. Actually, Rob Lefkowitz was a pioneer of the discovering the multiple signaling pathways, while Brian Kobilko is one of the key persons establishing the structure biology of GPCRs that finally allowed us to use structure-based approaches when designing new GPCR modulators. So next, I would like to just shortly give you a general picture on the GPCR structure. It has a very characteristic 7 transmembrane helices indicated this green, and therefore it could be identified as the Magnificent Seven. These 7 transmembrane helices, and these helices are located in the cell membrane, and there is an extracellular side and an intracellular side of the GPCR proteins. The extracellular side is pretty much important just because ligand binding. And the upper part of the protein structure is dedicated to the ligand binding module that shows much higher diversity between the different GPCR families as compared to the lower part that is dedicated to downstream signaling. When GPCR binds a ligand, there are conformational changes, of course, and there are some GPCRs having very significant induced effect. However, the conformational changes in the ligand binding module is typically smaller as compared to the downstream signaling module, where the much larger conformational changes might occur when the signaling molecules, typically G proteins or beta-arrestin, could bind. Now focusing a little bit to the binding packets available in Class A GPCRs. Of course, these are not all the binding packets experimentally explored. However, I would like to show you just some typical binding packets available in Class A GPCRs. And you can see here that the binding packet is pretty large in most of the cases, and of course it is open to the extracellular space. And this is the way how the ligand can approach the binding site. As you see, the adrenergic beta-1, beta-2, or dopaminergic, or even histaminergic binding site, then you could identify that the ligands are located in the lower part of the binding packet, and this is basically identified as the orthosteric pocket, typically used by the endogenous agonist, noradrenaline, or dopamine, or histamine. There are two special cases here. One is rhanopsin, which basically has no further space above the orthosteric binding packet. And the other special case is adenosine A2A receptor, because in this case, the receptor has been co-crystallized with a ligand that is reaching not only the orthosteric binding site typically used by adenosine, but also the ligand is extending to the upper part of the binding packet that is basically empty in the case of the adrenergic receptors or the dopamine histaminergic receptors. Just for a moment, you could see that this is not completely true for the histamine H1 receptor, because here is a phosphate group co-crystallized in the upper part of the binding packet. So basically, the upper part of the binding packet, just above the orthosteric binding packet, is usually called the secondary binding packet, abbreviated as SBP, that is closest to the extracellular space, and this is typically located in the extracellular vestibule. Now I would like to show you an NV simulation published a decade ago, and that was a very significant achievement at that time from the DE Show group, that they studied the ligand binding to the beta-2 receptor. And here you could see that first the ligand approaches the receptor, binds to the SBP, the secondary binding packet, and then it moved to the orthosteric binding packet and stabilized there. So this is an indication that probably in the case of class A GPCRs, the secondary binding packet is a temporal binding packet or a low affinity binding packet as compared to the more stable and high affinity orthosteric binding packet. Michino and co-workers analyzed the sequence similarities observed in the orthosteric and the secondary binding packet, and they found that typically the sequence similarity is much, much higher in the orthosteric binding packet, as indicated by this green color, as compared to the much lower conserved secondary binding packet. So this is basically not unexpected, because the OBP is specialized to bind the endogenous ligands, and that is basically small aromatic amines, and therefore the binding environment should be somehow similar. However, in every and each receptor, there are differences in the secondary binding packet that might be a very good option to use these differences to generate bitopic molecules that binds not only to the OBP but also to the SBP with designed pharmacological properties that includes the affinity, the selectivity, the functional activity, and so on. So let's see how exactly we can explore the secondary binding packet. As I already mentioned, the OBP provides a stable and primary binding site that is routinely screened experimentally. However, at least in the case of the class A receptors, the SBP is basically a temporary or metastable binding site that could not be assessed very routinely with experimental techniques, but of course this is available for virtual screening. So therefore we adapted a protocol to screen OBP and SBP. In the case of the OBP screening, we use the traditional experimental technique to identify an OBP binders, as indicated by this blue triangle. And then the OBP binder hits were docked into the GPCR structure to the orthosteric binding site, and we developed a protocol to dock different ligands to the SBP, to the allosteric side available in the extracellular vestibule in the presence of the already confirmed orthosteric ligand. So let's see the protocol in details. So first we screen the physical library of potentially OBP binding fragments, and we identified those fragments that were experimentally confirmed as OBP binders. Next, we compiled a virtual fragment library for potential SBP binders, and we dock the SBP fragment library in the presence of the experimentally validated OBP binder. So here you could see how an OBP binder occupies the orthosteric binding packet, and then we fix this binding mode over here and dock the SBP library to the extracellular vestibule, actually to the SBP, and identify those compounds that fit very well to the SBP in the presence of the OBP binder. Finally, we link the best combinations, like here, and these are the bitopic ligands we designed, and they are available for synthesis and, of course, testing. So let's see how this approach works when controlling the selectivity from the SBP. And here I have 3 examples. One for the dopamine D2, D3 receptors. I would add a little bit more details for these receptors, but I would like to show you 2 further examples. The other one is the serotonin 1B2B receptor, and what is more interesting, because these are not related receptors, these are the histaminergic H1 and the muscarinic M1 receptor. So let me start with dopamine D2, D3 receptors. At the time of the study, we had basically one crystal structure for the dopamine D2 and D3 receptors. So let's start with the dopamine D2 receptors that was co-crystallized with risperidone, that is a known antipsychotic compound. And actually this compound is a bitopic compound, because you can see here that the OBP binding part that is actually an aryl piperidine fits to the orthosteric binding site of the dopamine D2 receptor, and the SBP part, that is a bicyclic structural moiety, this extends towards the secondary binding packet as indicated here. In contrast, for the D3 receptor, we had only one structure at that time, and that structure was co-crystallized with ethylchloride. That is basically a dopamine analog in terms of the aromatic ring and also the basic nitrogen that is necessary for the high affinity binding to the aminergic receptors. Here you could see that ethylchloride fits only the orthosteric binding packet. If you compare the amino acid residues forming the orthosteric and the secondary binding site, then you could see that in the case of the OBP forming residues, basically there is no difference between the 2 receptors. And very few and only difference indicated in this figure is focused pretty much to the SBP region. And we would like to utilize this small difference to design compounds with significant D3 binding preference. So let's see how we did it. So as mentioned, first we screened a library of small fragments, typically differently substituted oryl piperazines and oryl piperidine fragments. And the reason is very simple, because these small fragments perfectly fit to the binding pharmacophore of dopamine, represented by the aromatic region and the basic nitrogen I already mentioned. When screening this library, we identified the best oryl piperazine and oryl piperidine fragments, and then we dug these fragments into the OBP. Next, we used a library of diverse small fragments that were dug into the SBP in the presence of the best binder oryl piperazines and oryl piperidines. And we identified the best combinations. So here you could see the results we obtained from this study. That was the proof of concept for the D2-D3 receptors. And you could realize that basically we identified the oryl piperazine or oryl piperidine moieties that fits very well to the OBP packet, and forming the very important interactions, the characteristic interactions between the basic nitrogen and the aspartate residue available in the third transmembrane helix. Next, we selected, in this case, this oryl piperazine that was fixed at the orthosteric binding site, and we dug the small library of potentially SBP binders here, and identified different cyclohexyl that fit very well to the SBP, and forming some interesting interactions. One I would like to mention is a hydrogen bond that formed with the glutamic acid residues available in the second transmembrane helix, and also some further interactions with the serine and threonine residues available in the seventh transmembrane helix. So next, we combined the OBP and SBP binding fragments with a linker and synthesized a couple of different compounds. On this slide, you could see the effect of the secondary binding packet binders, and I would like to show you first this OBP binder, that is the 2-methoxyphenyl piperazine, that originally showed a D2 preference. When we attached the SBP1 fragment to the basic nitrogen here, then a very small D3 preference was observed. However, when changing the SBP fragment from SBP1 to SBP2, that is pretty much similar, just the terminal nitrogen is demethylated in the case of SBP2, then we were able to improve the D3 preference significantly. Okay, let's see another OBP binding fragment, and this is the 2,3-dichlorophenyl piperazine that shows some minor D3 preference alone. And when combining the SBP1 and the SBP2, we were able to improve the D3 preference significantly. And something similar observed for the third OBP binder. This is the trifluoromethyl cyano-phenyl piperazine that shows some D3 preference. However, we improved significantly when extending the OBP fragments towards the secondary binding packet. So let's see what we can understand from structural studies when docking these compounds into the binding site of the D2 and D3 receptors. So first, I would like to show you that basically the orientation of the OBP fragment is basically the same. However, in the case of the D2 receptors, the cyclohexyl ring forms this favosteric contact with residue that is different in the D2 and the D3 receptors. Something similar happened at the other part of the SBP binding moiety. Basically, the terminal free amine forms a strong hydrogen bond to the glutamic acid available, the second transmembrane helix. And you can see here that there is another difference located pretty close to the SBP. And this difference is also responsible for the D3 selectivity we observed for this set of bitopic compounds. So at the end of the study was finally the identification of a new SBP pharmacophore. And let me tell you a little bit about the traditional antipsychotics and the general pharmacophore available for these compounds. So it is not unexpected that in the case of the OBP binding packet, you could see that there must be something aromatic, something hydrophobic here that mimics the dopamine's aromatic ring and forms interactions with the TM5 and TM6. And also, it is connected to the oryl piperazine moiety here, and the basic nitrogen is forming the hydrogen bonding interactions with the aspartate residue available in TM3. Up to this study or these results, it was considered that the other part of the molecule should contain something similar, a bicyclic or a monocyclic ring that should be located at the SBP and forming some interactions with TM1 and TM2. So here you could see two examples. One is aripiprazole that contains the oryl piperazine moiety here, and it is connected with a bicyclic ring that occupies the secondary binding packet. Something similar could be observed for risperidone. We have already the X-ray structure and already discussed that the oryl piperazine moiety here is extended again with a bicyclic structure that reaches the secondary binding packet. However, what we see here that the cyclohexyl derivative I already discussed previously that locates in the very same secondary binding packet and forms specific interactions with the D3 receptor as compared to the D2. So therefore, this compound, actually a known drug, cariprazine, discovered by our team at György and Richter, is the only D3 preferring antipsychotic as compared to the other D2 preferring compounds like aripiprazole and risperidone. Next slide. I would like to show you cariprazine, Vraylar in the US and Reagila in the EU, containing the well-known OBP binding moiety and the new SBP binding moiety that is responsible for its D3 receptor preference that finally turned this compound as potential medication for not only for schizophrenia but also other psychiatric diseases. So here in this figure you could see the team responsible for cariprazine discovery and development, and you could see also a younger edition of myself just standing in the second row. So the other example I would like to mention, this is the 5-HT1B2B receptors. Here we have two X-ray structures since ergotamine was co-crystallized with both 5-HT1B and 2B receptors. And it was very interesting to see from the X-ray structure that actually there is a methionine residue located differently in the 1B and 2B receptors that is responsible for the different action of ergotamine on these two receptors. If we compare the sequential composition of the OBP and the SBP, then here you could see that the OBP is basically similar, and the most different region of the sequence is located in the SBP, and that would help us to design specific or selective molecules for the 5-HT2B receptor. So let's see how we realize that, and I would like to start with the well-known compound LSD that binds to both receptors orthosteric binding site. LSD showed 10-fold preference for 5-HT1B receptor. When we extended LSD towards the SBP using ergotamine, then we did not see any preference between 5-HT1B and 2B receptors, you can see here. However, if we change the pretty complex SBP binding moiety of ergotamine to these simple fragments indicated as SBP-S1 and SBP-S2, then we achieved 35-fold and 26-fold preference for 5-HT2B. More interestingly, I would like to mention this compound, SBP-S2, because you could see here that as compared to the LSD, this compound showed activity only for 5-HT2B and 5-HT7, and therefore, just designing a suitable structural moiety for the SBP of the 5-HT2B receptor, we were able to achieve a very different pharmacological profile for this compound as compared to the multiple acting LSD. The third example I would like to mention, this is the histaminergic H1 and the muscarinergic M1 receptor. So here we started from a well-known observation, a clinical observation, because early antihistaminergic compound unfortunately showed some significant muscarinergic activity, and that is basically responsible for their unwanted side effects. So therefore, our objective was to improve the histaminergic selectivity, extending the OBP binding antihistaminergic compound towards the SBP. So here you could see that we started from this known drug that is bound to the orthosteric binding site of the H1 receptor and showed only 7-fold preference over the M1 receptor, and that is problematic just because of the very narrow therapeutic window of this compound. However, if we extend these molecules from the basic nitrogen, attaching different SBP binding moieties like SBP1 and SBP2, they were able to improve the H1 preference up to 128-fold. That is really a significant improvement. And interestingly, we found that this polar substituents, the polar moieties of the SBPs fits very well to the phosphate binding packet of the H1 receptor. So next, I would like to show you an example that we can not only control the affinity and the selectivity of bitopic ligands from the secondary binding packet, but more importantly, we can modulate the functional activity and the functional selectivity from the SBP. Why it is interesting? Because, as mentioned, the ligand binding packet of these receptors, these aminergic GPCRs, are located at the upper third of the transmembrane region. And that is located about 30-35 angstroms far from the signaling interface. And that's why, if we could realize that, that would be a true remote control action when modulating the functional activity. So just let me very shortly summarize you the GPCR signaling pathway. The most well-known is the G protein dependent pathway. That is a negative coupling to the adenylyl cyclase that increases the amount of the cyclic AMP and activates different protein kinases, and finally results in a cellular response. And there is another signaling pathway, and that is the G protein independent signaling pathway that is assisted by beta arrestins. That is a positive coupling, and then the beta arrestin's action is negatively coupled to the AKT and also the GSK3, and that finally results in different cellular responses. So we investigated both of the pathways, the G protein dependent and the independent signaling pathway in the case of D2 and D3 receptors. So let me first show you this RBP binding moiety when R group is simply a hydrogen. So this compound, compound 1, when there is no SBP binding moiety attached, proved to be an antagonist, not a very strong one. The PIC50 is around 5. Okay, when we attached an SBP binding moiety to the R group here, to the basic nitrogen of the piperazine moiety, we turned the original antagonist to partial agonist. And simultaneously, we improved the potency significantly, more than 2 orders of magnitude. Interestingly, however, when we changed the SBP binding moiety from SBP1 to SBP2, that is structurally pretty similar, but we again received an antagonist with pretty much the same potency as found for the partial agonist. So this is a clear indication that from the SBP, we can modulate the original functional activity, and the functional activity is depending on the structural moiety bound to the secondary binding packet. Just another example, the 2,3-dichlorphenylpiperazine, that is a partial agonist. It is actually much higher, having much higher potency than the previous one. And if we attach SBP1 that change antagonist to partial agonist, we can observe something similar because the partial agonist was turned to be a full agonist, and some improvement in the potency was also observed. However, if we change SBP1 to SBP2, that turn back the partial agonist to antagonist, here again, some significant decrease in the Emax value was observed, and this efficacy indicates that this is again a partial agonist as the starting point, but with improved potency. Okay, so it seems that from the SBP we can modulate the G protein signaling and also the potency of the compounds from the SBP. But what is the situation with beta-arrestin signaling? So if you see here that independently, whether we have or whether we have an SBP binding moiety or in the absence of the SBP binding moiety, we were not able to change the beta-arrestin signaling, since this compound without any SBP extension was an antagonist, and it has not been changed when attaching different SBP moieties. However, there was a significant improvement in the potency, and that is dedicated to the SBP extension here. And something similar happened for the other OBP binding moiety. It turned to be a partial agonist, but we were basically unable to turn this compound to full agonist despite of the SBP extensions applied previously for changing G protein signal. So therefore, we can conclude that we can manipulate G protein signaling specifically, selectively from the SBP while keeping the beta-arrestin signaling intact. So what would be the structural basis of this type of functional modulation? And here you could see the result of our MD simulations. So the first one as the one of the bitopic compounds that is actually a partial agonist, and it was modeled in the active conformation of the D3 receptor, and the other one is the antagonist that was modeled in the inactive conformation of the D3 receptor. So here you could say that we did not observe any specific interactions formed between the SBP binding moieties and the SBP package, the transmembrane helices here, because we observed pretty much the same interaction as a water-mediated hydrogen bonding of this amide nitrogen to the glutamate residue available in the second transmembrane helices. And this is basically the same for the antagonist. So the SBP context simply could not rationalize the different functional activity we found. However, when we analyzed the orientation of the OBP binding moieties, and here you could see in this insert, then the orientation of the OBP binding moieties are pretty much different in the case of the partial agonist and the antagonist. And the interaction that is responsible for the receptor activation, and this is the serine residue used by the dopamine itself when activating the receptor, is only contacted by that orientation, what we found in the case of the partial agonist, because in the antagonist conformation, we simply could not obtain similar interactions. So therefore, we can conclude that although we changed only the SBP binding moiety here, finally these changes reoriented the OBP moiety, and this is the mechanism of the remote control, how we were able to adjust the functional activity of the compound from the SBP. So we use the OBP as in the case of the endogenous ligands. So at the very end of my talk, I would like to just shortly mention another option. We can modulate binding kinetics from the secondary binding packet. And here we can go back to the antipsychotics again. Here you could see aripiprazole, and the other one is, of course, cariprazine, Vraylar here. And you could realize that these two compounds share pretty much the same OBP binding moiety. However, as already pointed out, the SBP binding moiety is different, because in the case of aripiprazole, that is the very conventional bicyclic SBP binding moiety, and here we have the non-cyclic, the acyclic SBP binding moiety. So as you see here, when measuring the binding kinetics on the D3 receptor, then we found that for aripiprazole, both monophasic and biphasic fits were okay for the measured time points. In contrast, however, in the case of cariprazine, we found that it is the biphasic fit describes better the cariprazine binding kinetics as compared to the monophasic fit. So this suggests that the mechanism of aripiprazole binding should be different from that of the cariprazine binding. So therefore, we performed special molecular dynamic simulations and simulated how aripiprazole is approaching the binding site. And it was very obvious that aripiprazole reached the orthosteric binding packet relatively straightforwardly and fit very well to the OBP packet, forming the characteristic interaction of the basic nitrogen with the aspartate residue in the TM3. And there are some specific interactions formed between the bicyclic system and serine and isoleucine residues available in the second extracellular loop. In contrast, for cariprazine, we were able to identify an SBP binding mode here. So you could see that cariprazine does not reach the OBP straightforwardly. However, there is an SBP binding position here when the basic nitrogen forms interactions with another serine of the ECR2, and the SBP binding moiety is located between TM1 and TM7. When the time goes on, cariprazine finally reaches the OBP and forming the characteristic interaction and stabilizing its binding mode within the orthosteric binding packet. So this is only a highlight that these two compounds, differing only in the SBP, shows very different binding kinetics. That is a clear indication that we can modulate the binding kinetics from the secondary binding packet. So at the end of my talk, I would like to summarize what we found. So the good news is that there are multiple options for the functional modulation of GPCRs from the secondary binding packet. But unfortunately, at least for the class A receptor, the SBPs are mostly transient or low affinity binding sites. So therefore, we could only realize this concept using bitopic compounds. The good news is, however, that the fragment-based approach I introduced today is very effective to design and to build bitopic compounds with desired pharmacological properties that would include the potency, selectivity, functional activity, functional selectivity, and binding kinetics. So at the end of my talk, I would conclude that the Magnificent Seven could be approached as a LEGO-like approach, just building the bitopic compounds within the binding site of the receptor on that way to produce bitopic compounds with the desired properties. At the end of my talk, I would like to acknowledge the contribution of my colleagues here at the Institute, also at György Richter, and also the collaborators at the Vrije University Amsterdam, University of Santiago de Compostela, the Heine and the Marburg University of Germany, and the Nottingham University in the UK. And thank you very much for your kind attention.